Chapter 1 provides us with an overview of biochemistry. Kind of lets us take a look at the pieces of chemistry and the pieces of biology and overall how these come together and why it's important to study biochemistry. So just to look at some of the goals for this particular chapter, we want to understand the definitions which will serve as the basis for biochemistry. We want to understand the classification schemes for organizing biological systems, understand and differentiate between the different types of intermolecular forces. So this is some of our chemistry coming in here. We want to define the building blocks of life. So we want to look at these individual molecules that serve as the building blocks. We want to understand the basic thermodynamic principles. So here we are taking thermodynamics, which we learn in general chemistry, and we're applying them to biological systems and explaining why processes occur. We will review pH definitions and look at why pH is so important in biological systems. With pH, we also want to understand buffers. So just in general, what buffers are and which buffers are important for biological systems. We also want to understand why water is essential for life. So first, let's look at what biochemistry actually is. And biochemistry is the study of chemistry, but it's the chemistry of life processes. So chemistry as it relates to biological systems. And so what we're doing here is we are studying the different types of these biological molecules and macromolecules that we will encounter. So we have molecules and macromolecules. Now, when we're talking about these molecules, we are talking about things like glucose, or glycerol. Okay. These are going to be low molecular weight compounds. Low molecular weight compounds. Okay. These are going to be our molecules. And often we're going to refer to these molecules as metabolites. Now these differ from the larger macromolecules, which are going to be our proteins and lipids, etc. These larger molecules that are involved in biological systems. And so we refer to these as biological macromolecules. And as we continue our study of biochemistry and we move through the material, we will take a look at each different class of biological macromolecule in a lot more detail. In the study of biochemistry, there are some basic themes which we look at, or which we will see throughout. So first, all living things make and use, so they're going to make and use the same types of biomolecules. So there are only certain categories of biomolecules that are incorporated here, and they're all going to use energy. All living things can be studied using methods of chemistry and physics, so we can look at biology through the lenses of chemistry and physics. And the fundamental similarities of cells leads us to speculate on the origins of life. So we can look at cells and how they operate, and we can look at the components in them, and we can tie this back to how life began. So just generally speaking, how does biochemistry describe life processes? Well, again, we have the same types of biomolecules, and we have this energy use that's occurring. And we know that biochemistry is multidisciplinary, okay, where we can look at the different areas of science and incorporate it into the study of biological systems. We can use results from many different sciences to answer questions, and these questions are regarding molecular nature of processes. So any biology class you have, you do go through a study of basic chemistry within those. And there are themes in terms of how cells will function, But remember that there are also differences in terms of cells. So we look at the fundamental similarities of cells and how that lets us see the origins of life. But we can also look at the differences in these cells and think about how life or how evolution occurred. Because there are some molecules that may play multiple roles, and there are other molecules that only participate in specific reactions. There are some reactions that take place in all cells, and there are other reactions that are only going to take place in specific types of cells. And we'll look at this as we move through the study of biochemistry. And so we mentioned that we can look at the fundamental similarities and differences in order to get some information about the origins of life. And so I think that most of you, or hopefully most of you, have seen this organization scheme in the past, this tree of life, which illustrates the distinct evolutionary paths that all arise from a common source. So we start with one common source and then branch out, and we can classify organisms based on different properties that we see. So we have eukarya, which are multicellular and unicellular, microscopic unicellular organisms. So this is going to range from anything from yeast to humans. So all organisms in this group will be referred to as eukaryotes. And remember that eukaryotes, let's give our little definition down here. Eukaryotes, these are going to be defined by the presence of a well-defined nucleus within each cell. So eukaryotes have a well-defined nucleus. And then we also have prokaryotes, which the prokaryotes we are going to see will include this archaea and bacteria. And these are going to be defined as, so these are our prokaryotes. These are going to lack a nucleus. But then we can further break these down, again, according to some of the properties that are present. So now comparing prokaryotes and eukaryotes in a little bit more detail, we can see in this table, here we are looking at the different organelles that are present, and then we can look at eukaryotes versus the prokaryotes. And so if we're looking at the nucleus, remember we said that eukaryotes are defined by the fact that they have this defined nucleus. Okay, there's a presence of a nucleus, whereas in prokaryotes there's no definite nucleus. Now there's still DNA that's present, but that is not separated from the rest of the cell. There's no defined nucleus to contain it. When we're looking at cell membranes or plasma membranes, that's going to be present in both prokaryotes and eukaryotes. If we look at the mitochondria, these are membrane-bound organelles. This is going to be present in eukaryotes, but there's no mitochondria in the prokaryotes. Enzymes for oxidation reactions will actually be located on the plasma membrane. Endoplasmic reticulum, again, those organelles are present in eukaryotes, but not present in prokaryotes. Whereas ribosomes will be present in both eukaryotes and prokaryotes. And then chloroplasts. Remember that chloroplasts in eukaryotic cells, these are present in green plants. But in prokaryotic cells, we do not have the presence of chloroplasts. So if there is photosynthesis, it's going to be occurring in, it's localized in chromatophores. Okay, special molecules that are going to be present to take care of that. So we talk about the origins of life, and we know that the Earth is the only planet that supports life, or we believe the Earth is the only planet that supports life at this point. So how did this come to be? Where did all of this originate? Well, you know that one theory out there, and you've all heard of this, is the Big Bang Theory. Okay. So the Big Bang Theory is the most widely accepted theory. Okay, and it tells us the origin of life. So this says that the early universe had very high temperatures, and that the average temperature of the universe has really been decreasing ever since because of expansion. And because of this, it has allowed for formation of stars and planets. So in this Big Bang Theory, all matter was originally confined to a very small space. And then an explosion occurred, and as the result of this explosion, matter started to expand with great force. So temperatures were approximately 15 times 10 to the 9th Kelvin, so very high temperatures. And that's very high, so the average temperature of the universe since that initial event happened has been decreasing over time. Now, in the earliest stages of the universe, when we're talking way back when Earth first started to form, the only elements that were present were hydrogen, helium, and lithium. Okay, there were very few elements that were present when it first started, those early stages of the universe. Now, other elements were later formed, and these were formed through thermonuclear reactions that occurred in stars, with explosions of stars and actions of cosmic rays outside the stars. So if we're looking at this timeline, you see that Earth, it was believed to be formed somewhere between 4 and 5 billion years ago. So Earth is about 4 to 5 billion years old. Now, the atmosphere that existed way back at the beginning is nothing near what the atmosphere is today. It was very different. Now, it was believed, the origins, we had very few elements present, other elements formed through those thermonuclear reactions, but there was very little free oxygen that would have existed in the early atmosphere. Now, remember that with the sun, everything there, that early on, there was constant ultraviolet light that was kind of irradiating and bombarding the Earth. And that ultraviolet light is what led to the idea of the primordial soup. And that primordial soup, okay, just thinking back to what that really was, basically this is the idea that molecules in cells arose from simple molecules. So that primordial soup, really, we were looking at water, some methane, maybe some CO2, some ammonia, some nitrogen, some hydrogen. So simpler molecules that were present, and because of the ultraviolet light and that constant bombardment, these were able to organize and form and start to make early building blocks of life. Now you can see here on this timeline, there's been a lot of change. So you had the Earth formed, really primitive atmosphere, started to form these microorganisms about 3.5 billion years ago. Cells with nuclei weren't formed until somewhere between 2 and 2.5 billion years ago. Then we started to get this oxygen atmosphere forming. Then we ended up with the macroscopic organisms, then the dinosaurs, and then human beings. So there's been this change in what's present over time. So if we kind of fast forward to living organisms as we know them today, living systems, we know, are complicated and they are highly organized. Okay, complicated and highly organized. And there are going to be many levels to this organization that occurs. We have compartmentalization within these systems, and even though they're compartmentalized, they function in collaboration. So when we're thinking about this organization, think about cells and organelles. We have cells, which kind of form organized structures, and inside those cells we have organelles that do different functions, and the organelles all function together to help the cells function. So this is just an example of that complicated and highly organized structure. Now, in terms of our function, or functional purposes, we'll discuss this greater as the semester progresses, but you've all heard that phrase that structure dictates function. Okay? And this is especially important in regards to these large polymeric biological macromolecules. Okay? These biological macromolecules that make us who we are. That structure is very important, and the way that that structure comes together will dictate how it works. In terms of the energy transformation, okay, so living organisms are actively engaged in energy transformation. Energy transformation is key for living systems. Okay, we know that the ultimate source of energy is the sun. Okay, that's our ultimate source of energy. Now, plants are going to capture energy from the sun, and that energy is going to be passed through the food chain. There are specialized biomolecules that will aid in the transfer of energy, and some of these, for example, specialized biomolecules, ATP, that's kind of our energy currency of the cell. And then we have these electron carriers, these high energy electron carriers that will come into play and we'll study as we move through different processes, like the NADPH. Now, in terms of self-replication, living organisms have remarkable capacity for self-replication. This is key in order to sustain life. Organisms are going to have mechanisms in place that allow them to make biological copies. So we have DNA replication that occurs. So we can apply this idea of this organization and levels of structure to the human body as we know it. So remember, we said that molecules and cells, we've got our molecules here, our atoms and molecules, that these kind of arose, the molecules and the cells arose from these simple molecules, like water, like oxygen, like hydrogen. And the simple molecules formed from combining these atoms together to lead to more complex molecules. Then you had some further organization into these macromolecules, which are going to be these biological molecules. So one example of a macromolecule would be proteins. Okay? We also, we're going to look at lipids, nucleic acids, many different biological categories of biological macromolecules that we're going to study throughout the semester. Then these macromolecules become organized within these organelles, which are organized within cells, and cells organize into different tissues based on the function, which organize into different organs, and then all of that is organized within the body system of the organism, or the human being overall. So one of the things just to note here is that as we move through our study, we are going to be looking at molecules that you have encountered in organic chemistry. We're going to be looking at the reactions that you've studied in organic chemistry. Here we're just going to show applications of those molecules, or we'll see that they're operating in a different context from what you would have studied them in your organic chemistry class. So taking a look at really the chemical foundations of biochemistry, these are all those functional groups that we looked at, or those compounds that we looked at in organic chemistry. And remember that organic chemistry, this is the study of compounds of carbon. And we saw that, or we will see that these carbon-based compounds are really essential to life. Carbon-based compounds are going to be the basis for all of these biological macromolecules as we move through. And within organic chemistry, you've studied these functional groups, which we know will impart physical or chemical properties to the different substances that we have. So it is going to be important to remember the different functional groups. These are relevant in biological systems. We're going to see them in different types of reactions. So you have your alkenes, your alcohols, your ethers, amines, thiols, aldehydes, ketones, carboxylic acids, esters, amides, phosphoric acid esters, phosphoric acid anhydrides. We are going to see these throughout our study of biochemistry. Okay? And the type of reaction that occurs in a biological system or in a particular process will be based on which of these classes of compounds or functional groups we're looking at. Some of these groups contain oxygen and nitrogen. And remember that these are among the most electronegative of the elements. Many of these functional groups are going to be polar, and this is all going to be important for when we start to look at reactivity of substances. So just to further illustrate the importance of these functional groups, here we can use, really, ATP is an example of what's going on. So you can take a phosphoric acid and an alcohol and react those together. Dehydration reaction occurring here, and you form an ester of phosphoric acid. You can have the phosphoric acid undergo reactions where you're combining. Here we have two of these phosphoric acid molecules coming together to form an anhydride of phosphoric acid. You can take the anhydride and an ester. You can esterify these things. We've got some ring structures here, some other molecules. But the functional groups are going to be present, and the type of functional group will dictate which type of reaction and which type of molecule will ultimately form. So carbon-based compounds are going to be very important, but there are also other elements that are going to be important in biological systems. So here we're just looking at the relative abundance of some of the important elements. Okay? So we're looking at abundance within organisms and then abundance in the universe. So this ranks these in terms of the most important or abundant to the least important or abundant. And you see that hydrogen, carbon, nitrogen, oxygen, sodium, magnesium, phosphorus, sulfur, potassium, calcium, manganese, iron, and zinc, these all have a relatively significant abundance within organisms. And just to give meaning to these numbers that we're looking at here, each of these abundance values that are given, it's given as the number of atoms that are present relative to 1,000 atoms of carbon. So for every 1,000 atoms of carbon that we have between 80 and 250 atoms of hydrogen that are present. Some of these, the carbon, hydrogen, nitrogen, oxygen, we will see these more in our macromolecules. That's why they have larger amounts that are present. Some of these substances, manganese, iron, zinc, we'll see these present in terms of kind of helping to facilitate enzyme reactions that occur. But all of these elements are important for biological systems. So going back to this idea of biomolecules, now that we've studied or we've talked about the different types of substances that are present and the characteristics of living systems, remember that we said we had small metabolites or small molecules, and then we had larger macromolecules. Now the larger macromolecules, when we look at these, we are really looking at proteins, nucleic acids, polysaccharides, and lipids, so carbohydrates. The larger macromolecules, these are going to be polymers that are made up of monomer units. So as we explore different biomolecules, we are going to look at the structures, and it's important to note that the structure will impart directionality within a lot of these. So if we're looking at in terms of monomer units that make up these biological polymers. When we're talking about proteins, the protein is the polymer. The amino acid is the monomer unit that makes it up. The nucleic acid is the polymer, and the nucleotides will make it up. The polysaccharides, or those carbohydrate structures, are the polymer, and monosaccharides will make up that polymer unit. And then the lipids, the polymer of the lipids, comes from glycerol and fatty acids. So as we mentioned, the structure of these different units and how they come together does impart directionality. And so when we're looking at directionality, we can see that things organize in a particular way, and that's going to be important as we move through our study. So here we're looking at DNA. Here we have a polypeptide segment, so this is a protein here. And then we have this polysaccharide chain or carbohydrate here. And it's more clear in this image for the DNA where we have labeled a 5' and a 3' end, which gives directionality. But we'll also see in these polypeptides, we're going to have a carboxyl and an amino terminus, which gives direction to that. And then with the polysaccharide chains, we're going to start to look at how these bonds form and in which way the bonds form, which provide us with directionality. So with our amino acids, as I mentioned, we have a carboxyl end and an amino end. The carboxyl end of one amino acid will end up being connected to the amino group of the next amino acid. So carboxyl end here connects with the amino group of the next. And when these two come together, we are forming a peptide bond, which we will actually see over here. Okay, we have peptide bond formation, and that allows the formation of our polypeptide structure. And this is going to serve as the basis for protein formation. With our DNA, again, we said that we had the 5' and the 3' end. Okay. So there are 5' and 3' ends of the DNA structure. The 3' hydroxyl group of the first nucleotide is going to end up forming a bond with the 5' hydroxyl of the neighboring nucleotide. And as you look at these types of polymer formation processes with the amino acids, with the DNA, you see that oftentimes we have elimination of water throughout the process. Now in carbohydrates, we have polysaccharides, which are going to be built, or these larger structures are going to be built by linking the first carbon of one monomer unit with the fourth carbon of a neighboring unit in this particular case. Now, different types of carbohydrates will come together in different ways, and we'll study that when we get to the chapter on carbohydrates. But here, this is showing first carbon and fourth carbon coming together to form a 1,4 glycosidic bond. This linkage would be a glycosidic bond. And the way they come together imparts directionality within the substance. And again, we see that water being eliminated here. And in the study of biomolecules, because we know that structure is so important and that will dictate function, we also need to talk about the types of interactions that are going to occur in biological macromolecules or biological systems. So just like we study in general chemistry, you have your bonding forces and your non-bonding forces. Sorry about that. Okay. So In general chemistry, we talked about these in terms of intramolecular versus intermolecular. So we're looking at intermolecular forces. So one of the first types of interactions we have are the covalent interactions that exist. Now the covalent interactions, you'll see these take slightly different names from what we have seen in the past. Covalent interactions are our bonding forces. These are going to be physical bonds that hold our molecule together. So these are holding all of these individual atoms together within this overall molecule here. These are the bonding forces, the covalent interactions. And the covalent interactions result from sharing of electron pairs. Because they are held together so tightly, they're sharing these electron pairs, they will require large amounts of energies to break them apart. And oftentimes, in biological systems, we are looking at these large ring structures. We'll see resonance become very important. And that resonance we're going to see not just ring structures, but anywhere you see a double bond, that resonance is going to help stabilize these structures. And those non-bonding or intermolecular forces, here we are calling non-covalent interactions. These are weaker than the bonding forces. Okay? These are going to be between the molecules. These are they're going to allow us to hold neighboring substances near one another. Okay? Again, same forces we studied in general chemistry. We'll just talk about them in a slightly different way. These weaker non-covalent interactions are crucial for biochemical processes. And in this particular textbook, it organizes them into 4 main types of interactions. We will have ionic interactions. Okay. And these ionic interactions, the charged group on one molecule can attract an oppositely charged group on the same molecule or on another molecule. The energy of this particular interaction or bond is going to be dictated by Coulomb's law, okay? Coulomb's energy. And we've seen this before, where in our terms, in general chemistry terms, we talk about this energy being proportional to the magnitude of the charges, so charge 1 times charge 2, divided by the distance between them. And that's what this is saying here. We're looking at 2 different charges and how much they're separated. So that means that the greater the charge difference, or the greater that product of the charge, and the shorter the distance, the stronger that ionic interaction is. So strength depends on relative charge of the species that are present. We also have our hydrogen bonds. Okay, so these are hydrogen bonds. They have they're very polar bonds, so they have a lot of ionic characteristic in terms of the interaction. It's based on partial charges existing in polar molecules. The hydrogen bond donor, remember, is a substance with a hydrogen that's attached to a nitrogen, oxygen, or a fluorine, but we don't really see fluorine much in biological systems. And the acceptor has to be a substance that has a lone pair of electrons, and that's really going to be another nitrogen, oxygen, or fluorine molecule. The more polar atoms will form stronger hydrogen bonds. We also will encounter van der Waals interactions. Van der Waals. These are based on fluctuating electron charge distributions about an atom. So that electron cloud becoming distorted as things are moving. Because a charge distribution results, neighboring atoms are attracted to one another. When atoms get too close to one another, if that charge is too similar or strong, then they're going to begin to repel one another. So the strength of these types of interactions will depend on relative size and distance between the substances. So the greater the area of contact between two molecules, the stronger the interaction that's going to occur. So there's some optimum van der Waals distance, which is going to give us the best attraction between the substances. And then finally, we can look at hydrophobic interactions, which are between two nonpolar and nonpolar molecules. These nonpolar substances are going to tend to prefer to associate with one another in water, and the strength of the interaction is determined by the degree to which the structure of water will be distorted when they come together. So we've looked at these weaker nonpolar interactions in the past. The longer the nonpolar portion of the molecule, the stronger that interaction becomes. These are going to be important, for example, when we have proteins. The nonpolar portions are going to hide in the core of the protein where they're away from surrounding water. So all those intermolecular forces that we studied in general chemistry are very important for biological systems. And of course, water is very important for biological systems. Most biochemical reactions will take place in water. So we know that water is essential to life, and it's essential to life for a number of different reasons. When we were looking in terms of general chemistry, the reasons that we described, we talked about the polar structure, which will help with interactions and intermolecular forces. We talked about the surface tension, the capillarity, the heat of vaporization. All of these properties were very important for life. Now, two especially relevant properties that we look at in biochemistry: the polar structure, again very important, and the cohesiveness of the substance. So the polarity of water is important because it allows for partially charged regions to interact with other molecules. And that bent structure, we know, is what imparts that polarity on the molecule. So that allows us to have that electric dipole forming, or that charge distribution. Now, the cohesiveness of water, that is what allows water to stay together, to interact with itself. And all of these are going to be important for biological systems. So this was kind of an introduction, or pulling the individual pieces from various classes that we've looked at, and just putting them together in context. Now, as we move through the semester, we're going to study these pieces in the larger picture of things, and in terms of very different biological macromolecules and how they function.